get a little noisy down there. Welcome to our lab. An underground mycorrhizal network is a fascinating symbiotic relationship between fungi and plants. Not only do they talk to each other by exchanging essential chemicals and water, some fungi also source their ability to degrade household pollutants such as crude oil and heavy metal. Scientists love them and did research on this sustainable practice. And we have some exciting things to show you today on measuring fungi spiral remediation. Here's Makeio to tell you more.
With the rise of urbanization and industrialization, pollution of soil with persistent organic pollutants is an increasing problem. Polycyclic aromatic hydrocarbons, or PAHs, the most abundant molecules in crude oil, contaminate soils and affect properties like texture, moisture, and conductivity. A sustainable solution to soil contamination is in situ remediation with fungi, better known as mycoremediation. Fungi, like Pleurotus, are particularly adept at decomposing PAHs. However, current methods of measuring mycoremediation, such as omics, are expensive and require laboratory settings.
Our product aims to provide a way to measure mycoremediation on-site through electrical signaling in mycelium. Our core methodology uses mycelium as a bridge to complete an electrical circuit, measuring how open circuit potential and resistance change when pollutants are introduced to the system.
The mycelial bridge utilizes the natural growth of fungi to form a biological conduit between two graphite nodes. Our design includes a two-chambered system separated by a small air gap, with each chamber containing a singular graphite node. By inoculating one chamber with mycelium spores, the spores naturally cross the air gap, completing an electrical circuit. This living bridge facilitates the measurement of biological signals that change in response to environmental pollutants.
To explain the inner workings of the experimentation, here is Netra and Shutong.
We did our experiment with agar and split it into 4 parts. We did the experiments to measure electrical signaling and also measured CO2 concentrations. We made our prototype first using 3D printing, graphite, filter bags, CO2 sensors, agar gel and mycelium. Agar gel will provide the nutrients for the mycelium. The first experiment was done with the 3D printed prototype. We prepared the agar with water after calculating its volume and autoclaved it for 15 minutes. Then we sterilized the solution in the Erlenmeyer flask with an aluminum foil covered on the sample. Then we decided to wait for a few days to solidify the gel. We found out that it solidifies very quickly when it cools down to room temperature. We observed that the mycelium was trying to move through the mycelium bridge, but it dried pretty quickly because of lack of moisture. Also, we realized it is better to use the silicone boxes because it is reusable and rewashable. So we switched it in the second experiment. For the second experiment, we used silicone boxes and graphite so that it was easier for us to autoclave them by preventing any contaminations from the environment. We put the same ingredients in the agar and poured this agar solution on both sides of the boxes and waited for it to solidify. After that, we injected the mycelium on the top of one of the sides of the gel. We inserted graphite into the gel this time with paraffin paper covered on the top to hold the graphites and avoid the mycelium from growing outside the box. Then we added water around silicone box to help mycelium grow with the moisture. We used CO2 sensor and taped it with the bag for measuring the growth of mycelium.
The third experiment was designed based on the second experiment, but what we changed was a variable in the silicone boxes. We put mycelium and agar in one side of the box and only the oil in another side of the box. But we did not see much of the change in a short period of the time. In the fourth experiment, what we did was the same process due to the second experiment, but we changed all the oil side into the oil and the agar side. So we also put the CO2 sensors and tape it into the filter bag, and we see much more growth than the third experiment.
But we did not stop here. We still got one more thing. Here's the next level FungiLink. With the same core methodology on getting a purified electric signal for measuring the living circuit living inside the air gap, the all-new unibody design with the introduction of the impulse measurement units and bacterial filter enable the FungiLink to go off the lab. The mycelium of the soil side chamber go across the bacterial filter, and for the first time you can access the real-world soil mycorrhizal network. The FungiLink is weather sealed and waterproof, And thanks to the in-device measurement units, the whole data collection process can be done remotely. This makes a huge difference from the invasive measures in the past, where scientists just insert electrodes into the mushroom, and have a big step forward from our previous generation of the FungiLink. Now let's talk material. While the device is not biodegradable, the materials selected are all recyclable and have ideal property to ensure the device functionality. The product is made to be used for an extensively long life cycle, just like any lab equipment. And the non-reusable part, including the agar and the bacteria filter, are all replaceable and harmless to the environment. Since the FungiLink is designed to be highly reusable, researchers can easily open and clean the two chambers and then inject the new agar gel and their mycelium culture. The way replacing the bacterial filter is also easy and requires little effort. Just open the white polypropylene cover and then cap it in. Once the chamber is filled with sterilized agar and mycelium culture, insert them to the spacer to create the air gap, and then properly insert the positive and negative node to the impulse measurement units. Although we are still on our way to finish the impulse measurement unit's electrical design, this concept of FungiLink marks a true evolution to access the data from underground mycorrhizal network. With the unibody in-device measurements and the ease of use, this is a step forward. Thanks for joining us today, and wish you all good luck on the Biodesign Challenge.